Hi team, we're going to talk about the citric acid cycle today, and I'm going to walk through some of the details that you will need to know. In fact, maybe more details than you need to know. And I'd like us to start way up here in the upper left-hand corner where we're drawing out the molecule pyruvate that has 3 carbons in it. And the first enzyme that we need is going to turn pyruvate into acetyl-CoA. Now this coenzyme A is not going to be attached to the molecule forever. It's just a coenzyme, and that's why I've drawn it in parentheses here. Now in this step, we're going to release CO2 because you can see pyruvate has 1, 2, 3 carbons, and acetyl-CoA just has 2. So this COO group is released as CO2. And in fact, I would say that this carbon is oxidized here, and we're going to need something then to be reduced. So we're also producing NADH, which you know is an electron carrier, and it's been reduced to carry those electrons. The enzyme that helps this happen is called Pyruvate Dehydrogenase. Now, the 2-carbon acetyl-CoA is going to join together with a 4-carbon molecule called oxaloacetic acid, OAA, and I'm going to draw that right here. So we've got the 4 carbons of oxaloacetic acid plus the 2 carbons of acetyl-CoA. They're going to come together to form a 6-carbon molecule called citrate. This is the same type of citrate that you have in your orange juice, citric acid, and this is where the pathway gets its name. This is citrate, and it has 6 carbons. The enzyme that helps us produce citrate from the 2-carbon acetyl-CoA and the 4-carbon oxaloacetate is called Citrate Synthase, because we're producing citrate. We're going to convert citrate into isocitrate just by rearranging the carbons. Really, I guess we're rearranging this oxygen to be down here. And then in the next step of the pathway, we're going to lose this COO2. It's going to leave as carbon dioxide, CO2. And if we're oxidizing carbon, we must be reducing something else, and that's NADH. And you know whenever we make NADH, we're going to be using a dehydrogenase enzyme. In this case, it's isocitrate dehydrogenase because we're starting with isocitrate, we're oxidizing CO2, and we're reducing NAD to NADH. The next molecule in the cycle is called alpha-ketoglutarate. And here's the structure of alpha-ketoglutarate. And I hope you notice here that isocitrate has 6 carbons, but alpha-ketoglutarate just now has 5 carbons because we released one as CO2. Alpha-ketoglutarate is going to get further oxidized to produce another CO2. That's going to be this CO2 down here that's released. And if we're oxidizing, we're also going to be reducing something. We create another molecule of NADH. If we're creating NADH, we must be using a dehydrogenase enzyme, and in this case, we're using alpha-ketoglutarate dehydrogenase. So the molecule that comes out of this is going to be a succinyl-CoA, which means we must be adding CoA into this reaction. Remember that CoA is not going to stay with this molecule forever. It's just there temporarily. And that's why I've put it in parentheses here. Now, adding CoA adds a lot of energy to this molecule. So we can break down succinyl-CoA to succinate, and in the process, create ATP. We're also going to lose the CoA right here. So here we have now the 4-carbon succinate, and we're going to rearrange succinate in a couple of steps to produce malate. And in the process of doing that, we're actually going to reduce a molecule of FAD to FADH2. So we've created another molecule carrying some high-energy electrons. I've drawn in here the structure of malate, and the next step is to convert malate back to oxaloacetic acid, OAA. And we're going to actually reduce malate. You can see we've got an OH group here, but in an OH group right here, but in OAA, we've lost that H. It's because we've reduced NADH. We've moved the hydrogen out to NADH, and now oxaloacetic acid is produced. I've drawn this as kind of a smushed up pathway, but you can see up here in the upper left, we've got the pyruvate bridge that converts pyruvate from the glycolysis cycle into acetyl-CoA that can enter right into the citric acid cycle. So that's often not thought of as part of the cycle, but a precursor step. And then I also want to show you that starting from about oxaloacetate right here, all the way through over to succinyl-CoA, I would call this the oxidation and reduction phase because we're releasing CO2. This is where we're releasing 2 molecules of carbon dioxide. And then the whole rest of the pathway, we have a 4-carbon, 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 4-carbon things. We're just rebuilding from the 4-carbon succinyl-CoA. We're trying to rebuild this starter molecule, oxaloacetic acid. So this half of the pathway is our rebuilding phase. And this first half of the pathway is our releasing CO2 phase. We briefly spoke in class that the citric acid cycle can't really turn off because we always need to be producing these NADH molecules and FADH2 so that we can ultimately use them in the electron transport chain. However, we can regulate the activity of this pathway depending on what the cell needs at any time. And so the 3 enzymes that are really important for regulation are isocitrate dehydrogenase, citrate synthase, and alpha ketoglutarate dehydrogenase. And several of the things that are important for regulating these enzymes include ATP, NADH, citrate, and ADP. So when we have lots of ATP, NADH, or citrate around, you can imagine that the cell doesn't need the citric acid cycle to work quite as hard. If there's a lot of these high energy molecules, or a lot of the citric acid cycle intermediates, then we can slow down the pathway. So I would call these allosteric inhibitors. They're not binding to the active site of the enzymes, but they're binding elsewhere on the molecule. That's what allosteric means. And they're going to inhibit the activity of the enzymes. Consequently, if we have lots of ADP around, we're going to want to speed up the pathway because this is a sign we need to build ATP. So we need to build some ATP. This is going to be an allosteric activator of these 3 important enzymes that can be regulated within the cell. Just a couple more things I'd like to point out to you about the citric acid cycle. Each of these carbon dioxides that are released, and we know that there's also a carbon dioxide released in the conversion from pyruvate to acetyl-CoA. All three of these carbon dioxides that are released come, at least initially, from this 3-carbon pyruvate molecule. So we're oxidizing completely all the carbons in pyruvate and releasing them into the environment of our body as CO2. So this is what we exhale as CO2. That's why all of our exhalation breaths are made of CO2. We've got to get rid of this CO2 somewhere. Okay? Another thing I'd like to point out to you is that this is really a cycle. So for every pyruvate that comes through, we're going to be running the cycle one time, but for every glucose, that means we'll have to run the cycle a second time because there are 2 pyruvates from each glucose. And this cycle really just keeps going and going and going and going inside the cell due to the fact that our bodies are in constant need of ATP. So we want to be constantly producing these electron carriers, FADH2 and NADH. So your body can constantly be burning the sugars that it's eaten to build up in the next cycle or in the next step in the electron transport chain to build up ATP from these stored electrons and to release that oxidized CO2 in the air we breathe out. So it's a cycle because we're doing this constantly. We're constantly regenerating the oxaloacetate so it can be added to the acetyl-CoA. All right, I hope this clears things up a little bit. Please bring any questions you have about the citric acid cycle into class or to office hours. Thank you.